So I want you to picture something for a second. Picture a teenager, right? It's the early 1970s.
Okay, setting the scene.
Yeah. And she's hitchhiking to Washington, D.C.
Hitchhiking, wow.
Right, hitchhiking to protest the Vietnam War. She's actually moved entirely out of her parents' house by this point.
Oh, wow. So really independent.
Incredibly independent. She's driving a cab just to pay the rent, and she's pulling these, like, late-night shifts, working as a cocktail waitress in this local jazz club.
Sounds like a pretty wild teenage life.
It totally does. But here's the crazy part. If I told you that this specific teenager, this cocktail waitress driving a cab, was going to grow up to fundamentally rewrite the laws of bioengineering, serve on a presidential advisory council, and actually win the Nobel Prize in chemistry.
You'd think I was making it up.
I would absolutely think you were making it up. You probably wouldn't believe me at all.
No, because it shatters basically every preconceived notion we have about, what a scientific pioneer is supposed to look like. We always expect this, like, quiet child with a pristine chemistry set who, never colors outside the lines.
Always does their homework, gets straight A's.
Exactly, the perfect track record.
But the real world is infinitely more interesting than that. So today, I want to warmly welcome you to a brand new deep dive.
And this is a really fun one.
It is. Our mission today is to trace this wildly unorthodox path. And we're using a highly comprehensive Wikipedia article on Frances Arnold as our sole source and guide for this.
Yeah, and her story is it's fascinating.
We are going to uncover exactly how this fiercely independent rebel basically hacked her way into the Ivy League and then pioneered an entirely new field of science called directed evolution.
Which is a huge deal.
A massive deal, because in the process, she fundamentally reshaped how humanity approaches manufacturing, medicine, and, renewable energy.
And I think the through line we're really going to see today, the core mechanism of her genius, honestly, is how her unique personality traits played into this.
You mean the rebellion stuff?
Right. Her absolute independence, this ruthless pragmatism she has, and just unbelievable resilience. It allowed her to do what, frankly, generations of chemists completely failed to do.
Because they were too stuck in the rules.
Exactly. She stopped trying to outsmart nature, and instead she figured out how to force nature to solve our most complex problems on this hyper-accelerated timeline.
It's so brilliant. So let's start right at the beginning, because her childhood really sets up this incredible psychological contrast.
Yeah, the background is key here.
So Frances Hamilton Arnold was born on July 25th, 1956, in Pittsburgh. She grew up in neighborhoods like Edgewood, Shadyside, and Squirrel Hill.
Classic Pittsburgh upbringing.
Right. But the environment inside her house was, well, it was incredibly structured.
To put it mildly.
Yeah. Her grandfather was literally a lieutenant general in the military, William Howard Arnold.
Okay, so heavy military discipline.
And her father, also named William Howard Arnold, was a nuclear physicist.
Oh.
I know. You are talking about a household steeped in not just military hierarchy, but the absolute rigid mathematical laws of nuclear physics.
That is a formidable legacy to be born into,
I can't even imagine the pressure.
Right, because when your family tree is built on military discipline and the strict absolutes of the atomic era, the expectation is total conformity.
Right. You fall in line.
Exactly. You are expected to respect authority, follow the established protocols, and just move predictably through the ranks of society.
But her response to that expectation was, total, unadulterated rebellion.
Which is where the hitchhiking comes in.
Exactly. As we mentioned, she's out there hitchhiking to D.C. to protest the government. She's living on her own as a teenager, driving a cab.
She's just completely rejecting the script.
And naturally, when you have a kid who is that fiercely independent, the traditional school system completely fails to contain them.
Because the school system is all about conformity.
Right. So at Taylor Alderdice High School, her grades were terrible.
Really, a future Nobel laureate.
Terrible grades. She had a massive number of absences, too. She just wasn't showing up. But, and this is a fascinating contradiction in the source material, despite her terrible grades, she achieved near-perfect scores on her standardized tests.
See, that tells us everything we need to know about how her mind actually works.
Oh, so?
Well, high absences and low grades in a bright student usually indicate a total refusal to engage with, rote compliance.
Like busy work?
Yeah, busy work. She simply wasn't motivated by the idea of sitting quietly at a desk just because an authority figure told her to.
That makes a lot of sense.
But those near-perfect test scores reveal this absolute powerhouse, of an intellect.
Right, she had the brain power.
The raw processing power was totally there. It was just entirely self-directed. She only engaged her mind when she felt there was an actual point to it.
And that pragmatic streak completely dictates how she handles college a few years later.
Oh, the Princeton story. This is my favorite part.
It's so good. So she decides she wants to go to Princeton University, which, by the way, happens to be her father's alma mater.
Which adds a fun layer of irony.
Definitely. Now, if you or I were high school students with a transcript full of absences and bad grades, we'd probably be terrified.
We'd be writing some groveling essay, right? Begging the admissions committee for a chance.
Exactly, trying to explain away the bad grades. But Frances Arnold, she looks at the admission system and just decides to completely outmaneuver it.
She games the system.
He applies for a major in mechanical and aerospace engineering.
Which is wild.
Right. And years later, during her Nobel Prize interview, she admitted exactly why she did it. She said it was simply and I'm quoting here, the easiest option and the easiest way to get into Princeton University at the time.
It is just a stunning bit of strategic thinking.
It really is.
Because in the 1970s, mechanical and aerospace engineering programs were likely really under-enrolled.
Right, they needed bodies.
And they were certainly desperate for female applicants.
Oh, absolutely.
So she didn't look at the Ivy League with this hushed academic reverence. She looked at it like an engineer looks at a system. She found the structural weakness, the path of least resistance, to get exactly what she wanted.
You know, it totally reminds me of watching a video gamer figure out a speedrun exploit.
Oh, that's a perfect analogy.
Right. Like a player realizes that if they just jump against a specific pixel on a random wall, they bypass the entire difficult boss fight and warp straight to the final level.
And that's exactly what she did with Princeton. She found the admissions exploit and warped right in.
But it makes me wonder, and I want to ask you this from a psychological perspective, does this early lack of traditional academic discipline actually work in her favor later on?
How do you mean?
Well, if you spend your formative years learning to perfectly appease authority and color inside the lines, how do you suddenly pivot to overthrowing scientific paradigms as an adult?
Oh, that is a phenomenal insight.
Right, because it seems like she trained herself early on to ignore the rules.
Yeah, and to disrupt a scientific field, you have to possess a fundamental skepticism of the quote-unquote proper way of doing things.
Because the proper way is just how it's always been done.
Exactly. In science, especially at the cutting edge, the proper way is often just an inherited habit. It's just how the last generation did it.
Which doesn't mean it's the best way.
Not at all. If your entire self-worth is tied to following the rules and getting an A+ from your professors, you are terrified of failing. You're terrified of looking stupid.
You play it safe.
But Arnold's teenage years proved to her that the rules were entirely flexible, that early independence completely inoculated her against the fear of doing things the wrong way.
Okay, so she speedruns her way into Princeton's mechanical engineering program. You'd think that once she hacked her way in, she'd put her head down and just, do math for 4 years to prove she actually belonged there.
But she doesn't.
No, that restlessness doesn't disappear at all. She's constantly zigzagging. While technically an engineering major, she starts loading up her schedule with economics, Russian, and Italian.
A real mix.
Yeah, she's looking way beyond the laboratory, envisioning a potential future as a high-level diplomat or maybe a multinational CEO.
She wanted to run things on a global scale.
And then, right in the middle of this Ivy League education, she just hits pause and takes an entire year off.
And she definitely doesn't use that year to just relax on a beach.
No, she travels to Italy, and she gets a job working in a factory that actually manufactures parts for nuclear reactors.
Which is just incredible.
Right. She's speaking a foreign language, working from the ground up in a manufacturing environment, literally getting her hands dirty, building the physical architecture of her father's field of nuclear physics.
It's experiential learning at its absolute extreme.
Totally.
She's not just reading about it, she's building a mental library of how the physical world actually gets built, not just how it's drawn on a chalkboard by a professor.
Right. She wants to see the actual nuts and bolts.
Exactly.
So she eventually returns to Princeton and graduates in 1979. But by then, the global context has completely shifted, and so has her focus.
Because of the energy crisis.
Exactly. This is the late 70s. We're dealing with massive oil crises, long lines at the gas pumps, and a sudden global awakening to the fragility of fossil fuels.
It was a scary time for energy.
It was. So she starts working at Princeton Center for Energy and Environmental Studies, focusing entirely on solar energy and sustainable power.
That macro-level focus is so crucial here.
Why is that?
Because she is developing a worldview centered on the biggest problem civilization faces. How do we power the globe without destroying it?
Right. And after graduation, she takes that worldview on the road. Her 20s serially read like a geopolitical thriller.
She was everywhere.
She really was. She's working as an engineer in South Korea. She's working in Brazil. She eventually lands at the Solar Energy Research Institute in Colorado, where she's actually helping to write energy position papers for the United Nations.
Which sounds like the perfect career track for a future diplomat.
It does. But here is where the causality of her life forces a massive pivot. You'd think she'd stay in international diplomacy to change energy policy.
But she hits a wall.
She hits a huge wall. She realizes that you can't just write UN papers about solar energy. Someone actually has to invent and build the microscopic chemical infrastructure to make green energy viable in the real world.
Policy isn't enough without the technology.
Exactly. So in 1985, she makes a move that is delightfully baffling. She enrolls at the University of California, Berkeley to get a PhD in chemical engineering.
Which sounds like a logical next step, until you realize one minor detail.
What's that?
She had absolutely zero background in chemistry.
None.
None whatsoever.
I cannot get over this detail. The graduate committee accepted her based on her sheer raw intellect and her mechanical engineering background, but they essentially said, look, you don't know any chemistry, so you have to sit in on the undergraduate classes.
That is so humbling.
Imagine being a worldly mid-20s engineer who has lived in Italy, Brazil, and Korea, who literally writes policy for the UN.
Sitting in freshman chem.
Yes. Sitting in freshman chem with a bunch of 18-year-olds learning about, covalent bonds for the very first time.
It takes an incredible lack of ego to do that.
Most people would be way too embarrassed.
Oh, definitely. But we have to look at the massive strategic advantage this actually gave her.
What was the advantage?
Because she didn't grow up in the chemistry discipline, she wasn't burdened by its dogmas.
Okay, what do you mean by chemistry dogmas?
Well, traditional chemists are trained to view molecules through a very specific, almost purist lens.
Like it's an art form.
Yes. They are taught to revere the elegant theory of how a bond forms, the mathematical symmetry of a molecule. It's very academic.
But she wasn't an academic, pure chemist.
No, Arnold approached chemistry as a mechanical engineer. She didn't care about the aesthetic beauty of a molecule at all. She only cared about what that molecule could actually do.
Form over function? Or wait, function over form?
Exactly, function over form. She viewed chemical bonds the way an engineer views steel girders and rivets as materials to build a scalable, practical solution to an energy problem.
It's essentially a liberal arts approach to the hard sciences.
That's a great way to put it.
And I actually want to pause here for a second and speak directly to you, the listener, because I think there's a huge lesson here.
It really is.
Think about your own life, right? Your career path, your hobbies. How often do we feel anxious or quote-unquote behind because we pivoted?
All the time. People feel like they're failing if they aren't on a straight track.
Exactly. Because we changed our college major or switched industries in our 30s or took some weird job that seemed totally unrelated to our ultimate goals, we are so conditioned to value the straight, predictable line.
But Frances Arnold's 20s proved that a diverse toolkit, this zigzagging, seemingly chaotic path, is exactly what allows you to see the solutions that the hyper-specialists are completely blind to.
Yes. Her perceived deficit in chemistry was actually her greatest superpower.
Absolutely. Because she arrived at the frontier of biochemistry carrying a toolkit forged in nuclear manufacturing, solar engineering, and international diplomacy.
That is quite the resume.
It is. And that unique combination is what led directly to the quote-unquote aha moment that would eventually win her the Nobel Prize.
So having established herself as a chemical engineer, she finally turns her attention to the ultimate biological machines, which are enzymes.
Right.
Now, let's build the foundation for this. For those of us who haven't taken a biology class in a few years, what exactly is an enzyme, and why is the scientific community so utterly obsessed with them?
Okay, so at the most fundamental level, an enzyme is a biological catalyst.
A catalyst meaning it makes things happen.
Right. It is a highly specialized molecule, almost always a protein that speeds up chemical reactions.
Okay.
If you think of a living cell as a bustling microscopic city, enzymes are the machines and the factory workers of that city.
They're doing all the labor.
Exactly. They digest our food, they copy our DNA when cells divide, they build muscle tissue, they break down toxins in our liver.
Wow, they do everything.
Everything. Without enzymes, the chemical reactions necessary for life would happen so incredibly slowly that life itself just wouldn't exist.
So they're essential.
Yeah.
And nature has spent billions of years evolving these microscopic machines to be incredibly efficient at their specific jobs.
Yes, but, and this is the crucial part, they are highly efficient under very, very specific biological conditions.
Like what?
Well, the enzymes in the human body, for example, have evolved to work perfectly at exactly 98.6 degrees Fahrenheit
Right, body temperature.
in a watery environment at a neutral pH.
But human industry has completely different needs.
Completely different.
Right, because if we want to manufacture pharmaceutical drugs or create biofuels or break down plastic waste, we need chemical reactions to happen in giant industrial vats, right?
Yes.
Sometimes at boiling temperatures or freezing temperatures or totally submerged in harsh, toxic organic solvents.
And a natural human enzyme dropped into a vat of industrial solvent would it would instantly fall apart.
Just melt,
It
basically.
would denature, yeah. It would lose its delicate shape and therefore lose its function.
So the big challenge for scientists in the 1980s and 90s was, how do we create new enzymes that can survive and perform in these harsh industrial environments?
Exactly. And the traditional approach that all the pure chemists were using at the time was called rational design.
Rational design.
Yeah.
Meaning they tried to use logic and reason to basically build a protein from scratch.
That's it. They would look at the incredibly complex three-dimensional structure of a protein on a computer screen, scratch their heads, and try to manually rewrite the genetic code to change the protein's shape and function.
Which sounds totally logical. Why didn't that work? I mean, it just sounds like standard engineering.
Well, it failed because of something called Levinthal's paradox.
Levinthal's paradox, what is that?
It explains the sheer mathematical terror of protein folding.
Mathematical terror, I like that.
It's true. So a protein is just a long chain of amino acids, right?
Okay.
And the way that specific chain folds up into a 3D shape dictates exactly what it does. But the number of possible ways a single protein could fold is just astronomical.
How astronomical are we talking?
It's estimated to be around 10 to the 300th power.
10 to the 300th.
Yeah.
Yeah.
That is more possible shapes than there are atoms in the observable universe.
Oh my God. So trying to look at a computer screen and logically guess which of those 10 to the 300th shapes will make an enzyme survive in an industrial vat is essentially a fool's errand.
Completely. You're flying blind.
You're just guessing.
Yeah. It was slow, it was agonizingly frustrating, and it mostly just ended in failure. We simply did not, and honestly, to a large extent, still do not understand the physical rules of protein folding well enough to design them from scratch like that.
And this is where Frances Arnold's pragmatic, path of least resistance mindset steps in.
This is her speedrun exploit for biochemistry.
Yes. She looks at all these brilliant chemists banging their heads against the wall of rational design, and she basically says, let's stop trying to be smarter than nature.
Right.
She realizes we are never going to solve Levinthal's paradox. So let's just breed the proteins instead.
And that is the beautiful essence of directed evolution.
It's so smart.
She realized that nature had already invented the ultimate optimization algorithm, evolution by natural selection.
Survival of the fittest.
Right. Over millions of years, random genetic mutations occur, and the environment selects the winners. But Arnold didn't have millions of years.
She wanted to publish papers now.
Exactly. So she decided to hijack that algorithm and put it on fast forward in her laboratory.
Okay, so let's walk through exactly how she achieved this, because the methodology is fascinating. And let's look specifically at her seminal 1993 experiment.
This is the one that really changed everything.
Right. So her goal was to take a standard enzyme called subtilisin E and force it to function in a highly unnatural environment, specifically an organic solvent called DMF, or dimethylformamide.
Right. And just for context, dimethylformamide is a really harsh synthetic solvent heavily used in chemical manufacturing.
What's it normally used for?
It dissolves plastics and synthetic fibers.
Oh, wow.
So a natural enzyme dropped into DMF is instantly destroyed. But Arnold wanted an enzyme that thrived in it.
Okay, so she takes the specific sequence of DNA, the gene that acts as the blueprint for this subtilisin E enzyme, and to mutate it, she puts it through a process called error-prone PCR.
Right.
Now, explain the mechanics of this to me, because PCR is a term we hear a lot now, especially after the pandemic, but error-prone sounds like she's making a mistake on purpose.
That's exactly what she's doing. So PCR, or polymerase chain reaction, is essentially a biological photocopier for DNA.
Okay.
It uses cycles of heat to separate DNA strands, and an enzyme called polymerase to build matching copies, basically doubling the DNA over and over again.
So you start with one copy, then 2, 4, 8.
Right, until you have millions. And normally you want perfect copies.
Right, you want the exact same DNA.
But Arnold actually wanted mutations, so she intentionally sabotaged the biological photocopier.
Wait, how do you sabotage a biological photocopier?
By altering the chemical environment of the PCR reaction.
Like throwing a wrench in the gears.
Exactly. For instance, you can add manganese to the mix.
Manganese, okay.
Yeah, and adding manganese makes the polymerase enzyme super sloppy.
Sloppy how?
It causes it to grab the wrong genetic letters when it's copying. So it might put an A where a C should be, or a G where a T should be.
So it's making typos.
Exactly, biological typos. Or you can intentionally unbalance the ratio of the chemical building blocks. The result is that instead of a million perfect copies of the gene, you get this massive library of millions of slightly flawed, randomly mutated genes.
Okay, so she then takes this massive library of mutated genes and inserts them into bacteria. And the bacteria act as microscopic factories. They read the mutated DNA blueprints and pump out thousands and thousands of slightly different mutated versions of the enzyme.
Exactly.
But here is the critical engineering problem. You now have a soup of millions of mutated bacteria. How on earth do you know which mutation actually works in the toxic DMF solvent?
Right, because you can't test them all one by hand.
That would take lifetimes.
So you need a screening mechanism. And this is where her brilliance really shines, because the screen she designed was incredibly elegant and entirely visual.
Let's dive into the mechanics of this screen. How did it work?
Well, she took Petri dishes filled with agar, which is basically a jelly-like nutrient broth that bacteria love to grow on.
Okay.
But she laced this jelly with two very critical ingredients. First, the harsh DMF solvent.
Right. Toxic stuff.
And second, a milk protein called casein.
Casein? Like the stuff that makes milk look cloudy?
Exactly. So she spread her millions of mutated bacteria all across these plates. Now, most of the bacteria produced useless enzymes that were instantly destroyed by the DMF, just as expected.
Because they couldn't handle the solvent.
Right. They just sat there and did nothing.
Right.
But if a single bacterium happened to possess a random mutation that made its enzyme stable in the DMF, that functional enzyme would seep out of the bacterium and begin to hydrolyze, basically, to chop up and digest the cloudy milk protein surrounding it.
Oh, and as it digests the cloudy milk protein, it creates a perfectly clear ring, a halo, in the jelly around that specific bacterium.
Yes. She didn't have to test a million bacteria one by one with complex chemistry.
Yeah.
She literally just held the Petri dishes up to the light and looked for the bacteria surrounded by the biggest, clearest halos.
That is amazing.
Those are the clear winners.
I absolutely love this. I use this analogy all the time, but this is the ultimate A/B testing, but it's running on living biology.
Totally.
Like, if you're a software engineer and you want to know what color the subscribe button should be to get the most clicks on a website, you don't sit in a room debating the psychology of colors for weeks.
No, you just test it.
Exactly. You launch a red button, a blue button, and a green button simultaneously to different users, and you just let the data tell you which one wins.
Which is so much faster.
And Arnold is doing that exact same A/B testing, but with the fundamental code of life. So she sees the bacterium with the biggest halo on the plate. What is the physical next step?
Well, she physically takes a microscopic loop. She scrapes that specific winning bacterium right off the jelly. She breaks the cell open to extract its winning DNA plasmid
And she throws it right back into the broken photocopier round 2.
Exactly. She takes the winner, mutates it again to create a brand new library of variations based on that winner, puts them on new plates, and looks for an even bigger halo.
So it's iterative, generational optimization. The source material notes she ran this process for 4 sequential rounds. And the final result. After just 4 generations, she had bred an engineered enzyme that was 256 times more active in the unnatural DMF solvent than the original natural enzyme.
256 times.
That is just staggering.
It really is. To put that in perspective, imagine taking a gray wolf, right? And over the course of just 4 generations of selective breeding, you breed a dog that can run 256 times faster than the wolf.
Wow.
And she did that at the molecular level in a matter of weeks.
Now, I want to clarify something the source mentions, because it notes that she wasn't the very first human being to observe experimental evolution. It actually mentions a scientist named Barry Hall.
Right, Hall was doing earlier work.
So if others were already playing with this concept, what made Frances Arnold's specific methodology so revolutionary that she was awarded a 1/2 share of the 2018 Nobel Prize in Chemistry?
Well, it comes right back to her engineering pragmatism.
How so?
Previous scientists observed evolution happening in a lab, but usually as a way to study evolutionary theory itself.
Like pure science.
Right, like just watching how organisms naturally adapt to starvation over time, for example. But Arnold was the first to systematically, iteratively, and intentionally apply selection pressure to solve a highly specific, unnatural industrial problem.
She weaponized it.
Yes. She didn't just let biology adapt. She directed the evolution. She created a reliable, reproducible technological framework. She basically turned the messy process of evolution into a precise tool for synthetic chemistry.
And once you have a tool that powerful, you don't just stop at making an enzyme survive in a solvent.
No, not at all.
Which propels us right into the next era of her career. She begins pushing the boundaries to build molecules that nature never even dreamed of.
Yeah, because once the scientific community saw the 1993 subtilisin E paper, the floodgates completely opened.
People realized what she had built.
They realized directed evolution wasn't just a neat laboratory trick, it was the master key.
Initially, she used it to expand the physical tolerances of enzymes. She evolved enzymes to function in extremely high temperatures and extremely low temperatures. But why is that so important for industry?
Because industrial manufacturing often requires extreme heat to force chemical reactions to occur quickly.
Right.
If you can evolve an enzyme that thrives at, say, 150 degrees, you can drop it right into an existing industrial pipeline to catalyze the reaction cleanly, rather than having to use toxic heavy metals to do the exact same job.
So it's much greener.
Much greener.
But then she takes an even bigger leap. She stops just improving existing functions, and she starts forcing biology to perform entirely alien synthetic chemistry.
This is where it gets a little sci-fi.
It really does. The text gives some highly technical examples that we definitely need to unpack. It says she evolved a protein called cytochrome P450 to carry out reactions like cyclopropionation and carbene and nitrene transfer reactions.
Okay, let's bring this down.
Please do. First, what is cytochrome P450 in nature? What does it normally do?
So cytochrome P450 is a massive family of enzymes found in almost all living things. In humans, they are primarily located in the liver.
Okay, liver enzymes.
They contain an iron atom, a heme group, which is similar to what's in our blood, and their main job is to process toxins, oxidize them, and basically make them soluble so our bodies can excrete them.
So it's a natural biological detox machine.
Exactly.
And she takes this detox machine and evolves it to do cyclopropionation. Explain the organic chemistry of cyclopropionation to me in a way that actually makes sense.
Okay, so in organic chemistry, a cyclopropane is a ring made of exactly 3 carbon atoms.
3 carbons.
Right. Now, imagine trying to build a triangle out of really rigid plastic pieces. The angles are incredibly tight, right? 60 degrees.
That would be really hard to snap together.
Exactly. And because of those tight angles, the chemical bonds are under immense physical strain. They desperately want to pop open.
Like a loaded spring.
Exactly like a loaded spring. And because it's spring-loaded, cyclopropane rings are incredibly valuable in pharmaceutical drugs.
Why is that?
When a drug molecule containing a cyclopropane ring enters the human body, that spring-loaded energy helps the drug interact really powerfully with its biological target.
Oh, it makes the drug more effective.
Yes. The problem is, because they are so physically unstable, building a 3-carbon ring in a chemistry lab is incredibly difficult.
It must be dangerous, too.
It is. It requires toxic, explosive reagents to force those carbons into that tight triangle.
And I'm guessing there are virtually no natural enzymes on Earth that build cyclopropane rings.
Right. Nature rarely uses them because they're so strained.
But Arnold used directed evolution.
Yes, she used directed evolution on the cytochrome P450 enzyme, mutating the area around its iron core, and she trained this liver detox enzyme to start grabbing carbon atoms and forcing them into these highly unnatural, spring-loaded triangles.
Wow, she forced a biological machine to manufacture high-end synthetic pharmaceutical components.
She made biology do synthetic chemistry.
That is just mind-bending. And the carbene and nitrene transfer reactions the text mentioned, are they similar to that?
Very similar. Carbenes and nitrines are highly reactive, fleeing chemical species that chemists use to stitch complex molecules together.
Okay.
They are so incredibly reactive that they usually only exist for fractions of a second. But Arnold evolved enzymes to tame these wild chemical species and use them to construct completely novel carbon-nitrogen bonds.
She essentially gave biology a welding torch it never previously possessed.
That's a perfect analogy.
And she didn't just scale up the chemistry of single enzymes, she actually scaled up the complexity, moving from single proteins to entire cellular factories.
Right, zooming out to the whole cell.
Which brings us right back to her 1970s obsession with renewable energy.
The solar stuff.
Exactly. She started co-evolving entire biosynthetic pathways inside E. coli bacteria.
This is where we see the absolute mastery of her craft.
What exactly is a biosynthetic pathway?
A biosynthetic pathway is like an assembly line inside a cell. It's not just one enzyme working alone. It's a sequence of enzymes. So enzyme A builds a part and hands it off to enzyme B, which does some work and hands it to enzyme C to produce a final chemical product.
And the specific product she was targeting with this pathway was a molecule called isobutanol. Why is isobutanol the holy grail here?
Isobutanol is this incredibly versatile alcohol. It can be used directly as a substitute for gasoline in regular combustion engines.
Wait, really? Just straight into the engine?
Yeah. Or it can be easily upgraded into jet fuel, or even used as a precursor for a massive variety of plastics and pharmaceuticals.
So it's basically a green replacement for oil.
Exactly. If you can get bacteria to eat plant waste and efficiently pump out isobutanol, you have a renewable pipeline to completely replace petroleum.
That's incredible. But there was a biological roadblock in this E. coli assembly line, wasn't there?
A massive one.
What was it?
Well, to force the assembly line to run in the direction of isobutanol, the enzymes require a very specific cofactor. Think of a cofactor as the specific currency of energy, or like a specific type of chemical electricity that actually powers the reaction.
Okay, I follow.
The cofactor required by this specific pathway is a molecule called NADPH.
NADPH.
Yeah.
And what does the E. coli bacterium naturally produce?
It naturally produces a massive abundance of a totally different cofactor called NADH.
Wait, one letter difference.
Just one letter difference. The P just stands for a phosphate group, but chemically speaking, it's a total mismatch.
So the enzymes won't work.
The enzymes simply won't accept the NADH. The whole assembly line just shuts down.
Man, I love this engineering problem. It is exactly like importing a state-of-the-art manufacturing robot from Europe.
Oh, the voltage issue.
Exactly. You buy this expensive robot, you bolt it to the factory floor, you're ready to produce your product, but you suddenly realize the robot requires a 220-volt European power supply.
Right.
And your American factory only has standard 110-volt outlets. You have all the machinery, but the power supply is completely incompatible.
That's exactly what was happening in the bacteria.
Now, a traditional rational design biologist might look at this and say, okay, we need to completely genetically re-engineer the entire E. coli organism to stop making NADH and start making the European voltage, NADPH.
Which would be catastrophic.
Why?
Because if you try to fundamentally rewire the basic metabolic energy supply of a living cell, the cell will almost certainly die. It's just way too complex. You break everything else.
So Arnold, using directed evolution, approaches it from the exact opposite direction.
Right.
Instead of rewiring the factory's power grid, she just mutated the European robots.
Yes. She ran the enzymes through the error-prone photocopier over and over until she found a generation of enzymes that had evolved a physical shape capable of accepting the local 110-volt NADH power supply.
It is so elegant, it is incredibly efficient, and it completely bypassed a biological roadblock that had literally stalled the field for years.
She forced the enzymes to run on the fuel that was readily available on site.
And the innovations just keep compounding with her. She wasn't content just mutating existing single genes. She actually wanted to mix and match them.
Right, combining them.
She developed a computational method called schema.
And schema represents the perfect synthesis of her computational engineering background and her biochemical expertise.
What does it do?
It is a mathematical algorithm for structure-guided protein recombination.
Okay, the source explains that she uses schema to combine parts of different proteins to form functional chimeras that fold properly, and then she mutates them further. Now, the common analogy here is usually Legos, or like Mr. Potato Head.
Right, taking the arm of one toy and plugging it into another.
But I feel like that dramatically undersells the physical difficulty of protein folding.
It absolutely does. Proteins are not rigid plastic blocks with universal snapping points.
I don't just click together.
No, they are delicate, dynamically folded ribbons of amino acids. The better analogy is really automotive engineering.
Okay, let's hear it.
Imagine you have the high-performance engine of a Ferrari, right? And you want to drop it into the chassis of a lightweight Honda Civic.
That sounds dangerous.
Exactly. You can't just drop it in. The mounting brackets are totally different, the weight distribution is completely wrong, and the torque, the sheer power of that engine will literally tear the frame of this Civic apart the second you turn the key.
Right, you have to know exactly where the load-bearing stress points are before you try to combine them.
Exactly, and that's what schema is. Schema is the computational algorithm that calculates those molecular mounting brackets.
Oh, wow.
It analyzes the 3D structures of two completely different enzymes and mathematically identifies the exact crossover points.
The safe spots to cut.
Yes, the places where you can safely splice the genetic code of enzyme A into enzyme B without destroying the structural integrity of the final chimera.
So schema builds a stable Frankenstein chimera that won't just collapse on itself in the vat. And once she has that stable new structure, she unleashes the directed evolution algorithm on it to optimize whatever brand new function it now has.
It's basically a factory for new biological machines.
Wait, hold on. I have to interject here.
Yeah.
Because tracing this whole progression from making an enzyme survive in a toxic solvent to forcing it to build explosive carbon rings to rewiring an entire bacteria's power supply to pump out jet fuel precursors, aren't we crossing a massive Rubicon here, ethically, or at least safely?
What do you mean?
Well, if we are programming bacteria to run entirely unnatural synthetic chemistry, we are basically creating microscopic oil refineries that are alive.
Right?
What happens if these synthetic chimeras get out of the vat? Have we completely blurred the line between a living organism and an industrial machine?
That is the defining question of synthetic biology, and the answer is, frankly, yes, we have definitively blurred that line.
That's a little terrifying.
It is, but from Arnold's perspective, and really the perspective of the broader bioeconomy, blowing that line is the only way to save the planet.
Because of the green energy aspect.
Exactly. For the last 2 centuries, human manufacturing has relied entirely on extracting dead resources from the earth.
Pumping oil, mining coal.
Mining heavy metals and using extreme heat and toxic chemistry to forge what we need. Directed evolution offers an alternative. We can program living biology to grow the resources we need.
Oh, I see.
Instead of a belching petrochemical plant, you have a sealed vat of microbes silently and cleanly converting agricultural waste into jet fuel at room temperature.
That's a much better image.
The safety protocols for biocontainment are incredibly strict, but the environmental promise of transitioning from extraction to biological synthesis is unparalleled.
Which bridges us directly to her impact outside the laboratory. Because Frances Arnold didn't just publish these breakthroughs in academic journals and hope someone else figured out how to use them.
She patented her work.
Extensively. The source notes she holds over 40 U.S. patents. And she fully achieved her undergraduate dream of becoming a high-level executive and a global diplomat.
Her commercial footprint is massive.
Tell me about the companies.
Well, in 2005, she co-founded a company called Gevo.
Gevo.
Right. And Gevo's entire mission is based exactly on what we just discussed, commercializing those engineered E. coli and yeast pathways to produce isobutanol.
Turning renewable carbohydrates into sustainable aviation fuel.
And plastics.
Wow. And then in 2013, she co-founded another company called Provivi.
Oh, Provivi is brilliant.
What do they do?
They use directed evolution to tackle agriculture.
Okay, how?
Well, instead of spraying millions of gallons of toxic, broad-spectrum chemical pesticides onto crops...
Which kills everything.
Right, which kills beneficial insects and leaches into the water supply. Provivi engineered microbes to produce insect sex pheromones.
Pheromones, like the scent chemicals bugs use to communicate with each other.
Exactly. If you spray the crop field with these biologically synthesized pheromones, it completely confuses the male insects.
Wait, they can't find the females?
They cannot find the females. It's called mating disruption.
That's wild.
The pest population crashes naturally, without a single drop of toxic poison ever touching the food.
I just love how this perfectly closes the loop on her life. She was a 20-something undergraduate at Princeton in the 1970s, completely obsessed with solar energy and sustainable power.
Right, the UN papers.
And 40 years later, She is literally building the multinational corporations that are executing that green vision using a biological technology that she had to invent from scratch just to get there.
It's an incredible narrative arc.
And the corporate titans of the world absolutely took notice. In 2019, she was appointed to the board of directors of Alphabet Inc., which is the parent company of Google.
Right.
And she was only the third woman in history to hold that position.
Which is a remarkably telling appointment, honestly.
How so?
Well, you have one of the most powerful software and data companies on Earth placing a biochemist on their board.
It has to mean that Silicon Valley views biological engineering as the next great computing frontier, right?
It is the ultimate confirmation of that idea. Alphabet understands that DNA is, at its core, a programming language.
It's just code.
It is a 4-letter code that stores and processes information. We spent the last 50 years mastering digital binary code to manipulate information on screens.
Right.
The next 50 years will be defined by mastering genetic code to manipulate physical matter in the real world.
Wow.
And Arnold's expertise in hacking and rewriting that code is exactly what a visionary tech giant needs to navigate the next industrial revolution.
That makes total sense.
Right.
And her influence isn't just limited to corporate boardrooms. It reaches the absolute highest levels of global government.
Back to her policy roots.
Exactly. In January 2021, President Joe Biden appointed her as an external co-chair of the Council of Advisors on Science and Technology, which is known as PCAST.
PCST is a huge deal.
It is. And looking strictly at her stated goals from the source material, she really saw this as a mission of scientific advocacy. She said, and I'm quoting, "We have to reestablish the importance of science in policymaking. We need to reestablish the trust of the American people in science."
And operating entirely on the facts of her appointment, it represents the absolute pinnacle of her early ambitions to impact global policy.
Yeah.
She started her career writing position papers for the UN in Colorado, and now she is setting the scientific agenda for the executive branch of the United States.
It's amazing.
She has positioned herself at the exact intersection where cutting-edge scientific capability dictates national strategy.
It's such an incredible irony, though.
What is?
The teenage girl who hitched a ride to Washington, D.C. to protest the government in the 70s is now walking the halls of the White House to advise it.
Right.
She never abandoned her youthful desire to change the system. She just spent 40 years acquiring the most powerful intellectual tools imaginable so she could actually do it from the inside.
It is a soaring professional triumph by any metric.
I want to slow down here for a second.
Okay.
Because to truly understand Frances Arnold, we really have to look at the entire picture.
The personal side.
Yes. We've spent the last hour building up this image of an unstoppable titan of science, industry, and policy. But running completely parallel to these towering historical professional achievements is a personal story of staggering, profound tragedy.
Yeah, the personal timeline is really heavy.
And I think it is absolutely vital that we talk about the human being behind the science.
I agree completely, because when you examine the timeline of her personal life alongside her professional milestones, it puts her achievements into a deeply humbling, almost unimaginable perspective.
It really does. So she was married to a biochemical engineer named James E. Bailey, and they had a son together, James.
Okay.
But in 2001, James Bailey passed away from cancer.
Oh.
And just a few years later, in 2005, which if we look at the timeline, is the exact same year she is co-founding Vivo and revolutionizing renewable fuels. Frances herself is diagnosed with breast cancer.
She had to endure 18 months of intense medical treatment.
While running a lab and a startup.
Exactly. Balancing her very survival with her duties as a scientist and a mother.
She survived the cancer.
Yeah.
She had entered into a common law marriage with an incredibly prominent Caltech astrophysicist named Andrew E. Lange. They had 2 sons together, William and Joseph.
Okay.
But the tragedies continued. In 2010, Andrew Lange tragically committed suicide.
That is just awful.
It is. And the heartbreak didn't end there because, unimaginably, in 2016, her son William died in an accident.
Yeah.
We really need to pause and recognize the sheer emotional weight of that timeline.
It's almost too much to process.
During the exact decades that she was fundamentally changing the nature of chemistry, publishing paradigm-shifting papers, securing 40 patents, building companies, being inducted into all 3 national academies, and doing the grueling laboratory work that would ultimately win her the Nobel Prize.
Yeah.
She was enduring the devastating loss of 2 partners and a child, and surviving her own battle with cancer.
think about the logistical and emotional reality of her position.
No, so.
Well, as a principal investigator running a massive laboratory at Caltech, you aren't just sitting alone in a room thinking about enzymes.
Right. You're a manager.
You are managing dozens of PhD students and postdoctoral researchers. Their entire careers, their funding, their futures depend entirely on your leadership and your grants.
That's so much pressure.
To maintain that level of high-stakes academic leadership while navigating such profound personal grief, it requires a level of psychological fortitude that is, frankly, difficult to fathom.
When you look at a Nobel laureate standing on that stage in Stockholm, it is so incredibly easy to assume they had a smooth, paved, uninterrupted road to greatness.
We always assume they had it easy.
We assume they were afforded perfect peace and quiet to do their deep thinking. But her life proves that greatness does not happen in a vacuum devoid of pain.
Not at all.
The ability to keep going, to keep iterating in the lab, to keep pushing the boundaries of human knowledge while carrying that much sorrow. It makes her life an extraordinary testament to resilience. How does a person even walk into the laboratory after so much loss?
It speaks to a fundamental, unbreakable strength of character.
It really does.
And, for many scientists, the lab isn't just a workplace, it's a sanctuary.
A place to escape.
Or a place to focus. The act of scientific discovery, the process of identifying a problem, running an experiment, finding a solution, and physically moving human knowledge forward might have provided a very necessary anchor in a life that felt completely chaotic.
That makes a lot of sense.
The work itself is inherently optimistic. Directed evolution is literally about creating something new, optimizing, and moving forward. Perhaps that relentless scientific momentum was a mechanism for survival.
And what's truly amazing is that the grief didn't cause her to retreat from the world. She maintained a fierce, almost adrenaline-fueled embrace of life.
Yeah, her hobbies are intense.
If you look at her hobbies listed in the source material, she's an avid scuba diver, a skier, a world traveler, and a dirt bike rider.
Dirt bike riding.
I know. It's as if, having seen the fragility of life so intimately, she made a conscious mandate to experience the physical world as viscerally and intensely as possible.
She isn't hiding from the world. She is engaging with it at maximum velocity.
And crucially, she never lost her sense of humor or her perspective. In 2019, the year after she won the Nobel Prize, which is literally the highest honor a scientist can achieve, she made a cameo appearance, playing herself in an episode of the sitcom The Big Bang Theory.
I love that she did that.
And later, when asked about it, she jokingly claimed that appearing on a television sitcom was the single greatest accolade of her entire life,
Right.
ranking it above the Nobel Prize.
That humor is so revealing.
Isn't it?
It shows a spirit that simply refuses to be broken by tragedy, but it also shows a wonderful humility.
Right.
She has a deep reverence for the science itself, but a very healthy irreverence for the pomp and circumstance of her own prestige.
Which brings us to the profound core of this story. The rebellious teenager who refused to care about the rigid rules of high school grew up to be a Nobel laureate who refuses to take her own prestige too seriously.
It's a perfect circle.
It is. I want to turn this directly back to you, the listener. We've spent this deep dive exploring the complexities of molecular chimeras, error-prone PCR, and genetic mutations.
Heavy science.
Very heavy science. But the ultimate lesson of Frances Arnold's life has absolutely nothing to do with biochemistry.
No, it's universal.
You do not need to be a chemist to apply the principles of directed evolution to your own existence. Whether you are trying to build a career in a new industry, learning a complex new skill, navigating a massive life pivot in your 30s or 40s, or trying to find the strengths to overcome deep personal tragedies, the lesson remains exactly the same.
Which is?
You do not need a perfect, rational master plan from the start. You don't have to perfectly predict how the protein is going to fold.
You don't need to solve the paradox.
Right. You don't need a pristine, straight-line trajectory. You just need to try things. You need to launch your own experiments, hack the system when you have to, find your own halos, the things that actually work for you in your unique environment.
Find your halos. I like that.
Discard the strategies that don't serve you. Keep the ones that do. And bravely, relentlessly iterate.
That is the perfect synthesis.
Thank you.
And if I may leave the listener with one final mind-expanding thought based on everything we've explored today.
Please do.
We have seen how Frances Arnold could force evolution to create enzymes that function in toxic industrial solvents and synthesize renewable aviation fuels simply by nudging genetic mutations over a matter of weeks.
Right.
Now, right now, this technology largely operates in massive, specialized commercial vats and high-end university labs.
Very centralized.
Exactly. But what happens when this biological technology becomes massive, polluting industrial supply chains? Every city, or maybe even every local neighborhood, has its own vats of engineered, directed evolution microbes.
Localized biological factory.
Yes. Imagine microbes custom bred in an afternoon to eat the specific type of plastic waste generated by your city.
That would change everything.
Imagine local clinics armed with engineered enzymes that can generate bespoke, personalized pharmaceutical medicines on demand, tailored perfectly to a patient's genetics.
No more shipping drugs across the globe.
Right. For the entirety of human history, we have been passive observers of biological evolution, or at best, blunt extractors of its resources.
Sure.
But thanks to the unorthodox, rebellious genius of Frances Arnold, that era is over. We have the pen in our hands now, and we are actively writing nature's next chapter.
From a teenager hitchhiking to a protest to rewriting the genetic code of the planet, what an incredible journey. Thank you so much for joining us on this deep dive. Keep launching those experiments, keep iterating, and we'll see you next time.